download our Revise It Right revision app for hundreds of videos, quizzes, exam questions, tutor support, and so much more. This video is about monoclonal antibodies, and it is a triple only content. Now, monoclonal antibodies are made by cloning cells, and why they're so important and so useful is because they will only bind to one specific protein antigen. Okay, as we know when we learn about our vaccinations and our white blood cells produce something called an antibody which bind to a specific antigen. And why that's so useful you'll see in a few slides. But first you need to know how to make and how they produce these monoclonal antibodies. So first what happens is they get a mouse or a rat that looks something or nothing like this. Now that mouse or rat is injected with the chosen antigen. Okay, so this is the antigen where we are trying to get the monoclonal antibody to bind to. What happens is then the mouse or rat's lymphocytes, in particular their B lymphocytes, are taken from the mouse. And remember, it's these that produce the antibody that we're trying to clone. So these are taken from the mouse. Okay, so these here, these B lymphocytes, and I'm going to write they are taken from the mouse. Now it's important to understand that these lymphocytes do not readily divide on their own, but something that does readily divide and divide very quickly is a tumour cell. Okay, so we take a tumour cell that has been growing in a lab and we fuse that tumour cell. So we get here, we're going to get a tumour cell here, and what we're going to do is we're going to fuse that tumour cell with these B lymphocytes. Okay, so we're going to fuse that tumour cell with the B lymphocytes, like so. So fuse tumour cell with the lymphocytes. Now what happens is this tumour cell with the lymphocytes divide rapidly. Okay, so they divide rapidly, which produces lots of these tumour cells with the B lymphocytes. And that these in turn then produce lots of these monoclonal antibodies that we need. Okay, so that's how we produce lots of these monoclonal antibodies. Now, because monoclonal antibodies can bind only to specific chemicals or pathogens, they are very useful for lots of things. And I'm going to talk you through some of the things they're useful for now. And the first of which is in a pregnancy test. Now, it's important to understand that when a woman is pregnant, they have a hormone in their urine that stands or it's called HCG. Okay, and that's important to remember. So when a woman is pregnant, they have a hormone in their urine called HCG. Now we can produce monoclonal antibodies that bind to this HCG, and this is how pregnancy tests work. So here what I'm going to draw is a pregnancy test stick, and this is the part in which you wee on, or the lady that thinks she may be pregnant wheeze on. Now in this part here you have a blue bead or blue dye, like so, and attached to these blue dyes are the antibodies or the monoclonal antibodies that HCG will bind to. Okay, so in this part where the woman wheezes on, you have the blue beads which act as a dye, and you'll see why that's important in a second, and attached to that are the monoclonal antibodies that this HCG will bind to. Now, as well as the bit that the woman wheezes on, the test stick will also have something called a test strip. And this is the bit that will actually turn blue if the woman is pregnant. You know, you get those two blue lines. And this is the test strip here. And this contains also contains monoclonal antibodies that are stuck down. Now it's important they are stuck down. Now it's important these antibodies are stuck down so they can't move essentially. So when the hormone binds to it, they can't move. So you can see a nice blue line that hasn't moved. Now let's go through what happens then when the lady is pregnant. So the lady will obviously wee on the area where she will wee. And what will happen is the hormone, the HCG that's in her urine, will bind to these monoclonal antibodies. And what will happen is the bead, the urine will start moving up the test stick. Okay, the urine will start moving up the test stick, carrying the hormone and the beads with it. Now what will happen is the beads, okay, will now bind to these monoclonal antibodies that are on the test strip. So the blue beads will get stuck and essentially on the strip, and it will turn it blue. And that's where you'll see that blue line to say that they are pregnant. Okay, so the beads carried from here will stick to the antibodies that have been stuck down on a test strip and turn that strip blue. So what then happens if the lady is not pregnant? Well, the lady will still obviously wee on this part of the pregnancy test here, but there will be nothing to bind to these monoclonal antibodies here. The urine will still travel up the pregnancy test stick, and the blue beads will still travel up the pregnancy test stick as well. But these blue beads will not have anything to attach to because they don't have this HCG that's here. They will not attach to the monoclonal antibodies on this test strip here. So what will happen is they will pass the test strip like this, and they will not bind, and therefore the test strip will not turn blue. And that is how a pregnancy test works in using monoclonal antibodies. Remember to download our Revise It Right revision app, watch over 700 videos, answer 4,000 plus quiz questions, over 1,000 flashcards, 1,000 exam questions, worksheets, forums, and get help from qualified teachers, and so much more. The link is in the description. Now, monoclonal antibodies can also be used to treat disease. So for example, as we know, different cells in our body have different antigens on the surface. This is to allow our white blood cells to recognize them. So you can make monoclonal antibodies that bind to these specific cells. Now, why this is really important in treating disease, because cancer cells, for example, also have antigens on their cell membranes that aren't found in normal body cells. These are actually called tumour markers. So what we're going to do here is we're going to draw a cancer cell, and then here you can see that they have the tumour markers. Now what you can do is you can produce monoclonal antibodies that bind specifically to these tumour markers. Okay, so monoclonal antibodies can be produced that bind to these tumour markers. Now to make this even better, an anti-cancer drug, okay, can be attached. So an anti-cancer drug can actually be attached to these monoclonal antibodies. This might have a radioactive substance on, or a toxic drug, for example, which stops the cancer cells growing and dividing. So you can give the patient these monoclonal antibodies through a drip. They bind to the tumour markers on the tumour cell, but remember these have an anti-cancer drug that are attached to them that can bind to the tumour markers and then have an effect on the tumour cell, i.e. stopping it from growing and dividing. So this is a really useful way we can use monoclonal antibodies to treat disease. So another use of monoclonal antibodies is measuring levels of hormones or chemicals. So remember, our hormones or chemicals have specific antigens, and we can insert monoclonal antibodies into the body and they will bind to the hormones or the chemicals that you're trying to measure, and using that you can then detect or measure how much of a certain hormone or chemical a person has in their blood. Okay, so for example here we've got a hormone, and again it has a specific antigen. We know then if the monoclonal antibody attaches to that antigen, then we can use it to measure levels of that hormone or that chemical in the blood. Okay, so another thing we can do is measure the levels of hormones or chemicals in the blood. And finally, we can also use monoclonal antibodies in research as well. So for example, they can be used to locate specific molecules on a cell or in a tissue. So what you're going to use is you're going to get the monoclonal antibody that's going to bind to the specific antigen that you're trying to research, but you're going to attach a fluorescent dye to this monoclonal antibody. Okay, the antibodies will then be bound to the antigen, obviously with the dye on. And if the molecules are present in the sample is that you're trying to research or you're analysing, the monoclonal antibodies will attach to them like that happened here, and then they can be detected using the fluorescent dye. Okay, so for example, cancer cells that have been targeted by monoclonal antibodies that have a fluorescent dye attached, so we can see if there are cancer cells in the person, for example. Okay, so the antibody binds to the antigen on the molecule that's being located or researched, and then the dye can be detected from that. Now, monoclonal antibodies obviously have tons of uses and they sound amazing. Many advantages come by them. For example, treating cancer, researching particular diseases that could go on to find treatments for them. But you need to understand why monoclonal antibodies are not yet used as widely as expected. And a big reason for that is because of side effects. Okay, now, monoclonal antibodies actually cause more side effects than were originally expected. For example, they can cause fever, vomiting, and low blood pressure. Now, when these were first introduced, it was thought that they wouldn't carry much side effects because you can use them to target very specific regions, very specific antigens, molecules, chemicals, hormones, whatever. But they have given and been found to give more side effects than were originally thought, and that is why monoclonal antibodies are not yet used as widely as expected. So what I'd now like you to have a go at is answering these 3 questions here to test your understanding on the topic. So pause the video now to have a go. Okay, so question 1, how are monoclonal antibodies produced? Well, the antigen that you're trying to produce the monoclonal antibody for is injected into a mouse. The B lymphocytes produced from the mouse are extracted from the mouse and inserted into a tumour cell. These divide rapidly and therefore produce lots of the B lymphocyte, which in turn can produce lots of the monoclonal antibodies. The monoclonal antibodies for the antigen. Okay, so that's how they are produced. Give 3 ways monoclonal antibodies are used. Well, you can talk about the fact they're used in pregnancy tests. They're used to treat disease. They're used in research. And they're also used in labs to study different hormones and chemicals. And finally, give a disadvantage of using monoclonal antibodies. The big one that we talked about was the side effects. So for example, vomiting, fever, and a low blood pressure. So we've now looked at monoclonal antibodies and their uses. There are plenty of practice questions on the website to test yourself further, and the quiz as well. And as ever, if there's anything you didn't quite understand, please do get into contact with one of our tutors who will happily help.